I think we could go ahead and get started since we have a lot going on. So basically, I'll start off with some updates on our end, and then I will hand it over to our speakers for today. And so please feel free to use the chat, ask questions, raise your hand, however you'd like to interact with us today during this session. So first off, like I said, I just have some updates of events that we're going to be at and some information I'd like to share with all of you. So first up, the CMDKP will be at the ADA in 2026, again, June 5th through 8th in New Orleans. And we are very excited to be a part of the NIDDK Scientific Corner. We will be having our typical booth, and there'll be lots of other events that will be happening throughout the week. So please stay tuned as we have more that we will share once we get closer to the event. Just a reminder today that our webinars are going to be bi-weekly. That means it's going to be on the second and fourth Thursdays. So we are uptaking our frequency, and we're happy to have you here as we're hoping to get more science as we go throughout all of these sessions. Now, one other thing that I just wanted to note, so another project that we are working on is for the common metabolic or common fund data ecosystem. And so for this funding mechanism, there is a funding opportunity that we would like to bring to you in case you are a part of, the common fund. So essentially, we have an opportunity for pilot projects enhancing utility and usage of common fund data sets. If you were to scan this QR code, this will give you the information about the RFA. But essentially, this is closing in June, and there's a maximum of 200,000 award, and they're expecting 13 of the awards. But the idea is that there are very many data sets in the Common Fund programs, and basically this goal is to announce the availability of funding to demonstrate and enhance these data sets by coming up with hypotheses and coming up with new discoveries. So we'd love to have, members of our community apply for this if interested. And on another note as well, tomorrow we have our Common Fund Data Ecosystem public webinar series, and we will be presenting on behalf of the Knowledge Center, which is our team specifically. So if you want to hear new opportunities from our program, specifically site updates and can't miss events, we will be showcasing all of that tomorrow. So we really look forward to seeing you all there if you'll be there as well. So I will now stop and hand it over to our featured speakers, Noel Burke and Luca. So I'll hand it off to you, Noel.
Okay, thank you, Mackenzie. Excellent. I'm going to just set Luca up. Hopefully, I will set him up exactly how he needs. And I'm actually really exciting. Luca is a member of our KPN team, but also more broadly, he's part of the Flanic Lab. He's going to talk to you about some work. It's a bit of a departure from what we normally talk about at these sessions, which is kind of cool. And I think it's a sort of homage to kind of where we're headed as a Knowledge Portal Network team. And so that's why I'm going to set it up to kind of orient you from where we were and where we're headed. So you get a sense as to why we wanted to bring this to this community today and share it with you. So let me just get my screen up. Just give me a second. And again, as mentioned, Mackenzie mentioned, stop me if you have any questions and want to chat midway. But let me get my screen up. All right. There we are. And there we go. Okay. So can folks see that okay?
Yes,
Yeah.
thank you.
All right. So I think the sort of the approach, overall governing approach today is really about novel genetic and genomic resources for the exploration of disease mechanisms. That's really the approach that our team is starting to expand into. You know us for our genetics portals, right? But we also get to do a lot of other things with human genetics as our anchor. And our core competencies and the things we really think about when we build a portal. We also think about this when we build a resource, a scientific resource. And Luca's going to talk about a scientific resource in the form of a knowledge graph today. But they all are driven by some very basic principles, which is it's community driven. What I mean by that, it's like we think about what the community needs, what researchers want, and try to build to that very slowly and incrementally. And also the data we represent is community driven because they're the experts. They know their data. They know how they want it radiated into the world. And to do that, you have to build a flexible platform that can be changing over time and modular and adapt to the changing culture and society AI, read AI. So this is another sort of starting to move into that space where we are actually going to be more poised to adapt to that environment. And finally, we think about knowledge, on-demand knowledge, how we bring your scientific research context to knowledge anchored in human genetics and genomics. And the anchoring features to human genetics and genomics are genes, obviously, right? Anchored to traits and diseases. Those are all mitigated by mechanisms and processes, but they act through cell types. So this is this sort of like environmental circle. And then we have some signature products that sort of hang in that space, starting with the common metabolic disease knowledge portal, sort of our anchoring feature. We've expanded to all sorts of diseases for common disease with A2FKP. We have portals in specific tissues with MATKP and PANK-base. But as we've sort of been able to expand, we've also been to adapt to the world to think about processes. And the knowledge portal sort of moved into this world where we work with the common fund data ecosystem. So I'm going to quickly talk about that and then turn it over to Luca. But what we do in everything we do, human genetics is our backbone, namely because it's what we believe are observations of nature, which is great. There's something you can observe and run statistics on, and there's a statistically rigorous way by which you can measure them. And also, we believe and we know that drug success that uses human genetics is tied to better success long term, right? So this is proven by what we've done with our type 2 diabetes knowledge portal and the CMDKP in terms of we make it so that you can search your favorite gene, your favorite phenotype, and understand what we know about human genetics to provide genetic support to you, right? And that's all anchored on this assumption that there's a biological model, there's a variant, it impacts your disease or an organismal function, but there's all these things in between that happen to disrupt that, right? So our job is to figure out, we present how to get from here to there, but we have to pull together all the evidence sources to support how that's done. So over time, we hope to better leverage these different components, right? And we do this through resources and through that same core competency. So the core Common Fund Data ecosystem offered us an incredible opportunity. As you can see, it's an incredibly diverse, right? It's an NIH program, funds many, many resources, but it's come together in a coalition of an ecosystem of resources that you can see in the outer circle that have agreed to sort of come together, share data, share different tools, build together so that they can make it more useful for users to use that data, right? So the idea is there are centers at the center of this trying to like bring all the data together and make it more useful. We are the knowledge center particularly, but our goal here is to actually leverage the data and the knowledge in all these resources connected to our human genetics to enable sort of new scientific features, products, and tools. So this is the Knowledge Center. If you want to hang out there, it sort of gives you the global perspective of what the CFD is, what the different centers are, how it attached to different omics and entities, and then also more broadly, the centers that support all them. So you can sort of navigate that. That's cfdeknowledge.org. So
I just put the link in the chat for everyone as well.
perfect. Thank you, Mackenzie. So what this did was open up an entirely new world to us, which was super cool. Not only did we get great collaborators, but we got an entire new world of data to integrate with. One particular thing of the fact that many of these programs produce signatures or readouts, right? So metabolomics workbench, they produce metabolic signatures from basically different perturbations. Links perturbs every genes and observes perturbations. If you could take all those readouts, turn them into gene lists, whether it be by differential expression, which is the example I'm showing here, you can actually encode those and extract those gene sets, and they have meaning, right? They have meaning in terms of like their coordinated response to some perturbation or activity in all of these programs. And you can get at all that raw data. That's beautiful. So all that provenance is already tracked for you. So think about the substrate of 277,000 gene lists. How do we make them more useful? Well, to understand maybe pathways, mechanisms, or phenotype. And that's where human genetics comes in as a way to sort of anchor, or another way to sort of clean that set of, I think is a better word, all those gene sets to human phenotype to help us understand causality and maybe mechanism, right? So that's the landscape. So the anchor there is, again, 7,000 traits across common and rare disease. Again, community driven. We know these results are great. They're valid. These are the best resources in the world, in my opinion, to do them. So you got that. Then you bring in a model, which you've heard about in many different webinars and forums for us, right? To sort of connect all that. I'm not going to talk about that. Jason's the expert in that, but you've heard that before. That's the anchoring mechanism. So this brings together essentially a substrate for collaboration and validation for us. You've got your gene sets, got to run against the method, so you can link them to common and rare phenotypes. And these are provided on the Knowledge Centers of our classic workflows. So visualizations you're used to. You can type in your favorite gene, sort of see the results, kind of get a sense of why they're meaningful, but it's a little opaque unless you know exactly what you're looking for. So we've been thinking about other ways to make them more useful. And you're going to hear about two different directions. Really quickly from me about sort of how we've taken the underpinning resource under here, which Luca is going to talk about in detail, and sort of made it more useful from a context perspective, from a human like language query, plain language query. But then Luca's going to talk about how we've made it useful in a totally different context. And that's really our goal, right? To approach this world where we're going to be using AI agents to do a lot of the work for us. And we want to make sure that the data that these agents query is good. And so the structure of those is critical, right? So with that, um, this sort of feature, as I mentioned, sort of has, utility, but it has little meaning unless we can actually bring your context more to it. So the one way in which we've combined sort of these classic workflows is how do we bring like what you were thinking as a scientist, your potentially very detailed question, or I care about inflammation and heart disease. Think about those two things. If you could just bring those to a resource and have it query this resource, that would be valuable to you. So what we did was we encoded these extracted gene sets, connected them to human phenotype, and embedded them as vectors. And now we allow a UI for you, we call it CFD reveal, to navigate this. So that's tune in tomorrow to learn all about that. But that's what we call CFD reveal. And so this is the beginning of a way for you to use your context to navigate our resource. But what's underneath all this? And this is the cool part. Well, there's a lot that's cool, but this is really cool. What you're actually looking at and querying when you're running this is a genetically sort of anchored knowledge foundation or knowledge graph, truly. And it allows you to sort of explore this through either query or through query that technological query, for lack of a better term for me as a non-computer scientist, another way of investigating that whole graph. And there's a whole set of projects this opens up to us. And knowledge graphs are essentially, I would say, in my way, a language by which you can connect to other scientific resources to allow you to make queries, both in natural language through classic UIs, but also through Sparkle queries and all these different types of mechanisms that users can use. So that's what you're going to hear about next. And what's really cool is this is sort of like the backbone of so much of what we're going to be doing going forward. So we're hoping that we can expose this not only through UIs and classic mechanisms, but other access mechanisms. And by encoding it in certain ways, you can tap into all these other resources. So with that, unless there's any burning questions, I'm going to turn it over to Luca, who's going to tell you really about the details behind that.
Thank you so much. Let me try to share some slides. So I'm going to be talking about a few different things. Whoops. So that was a very good setup to give some maybe motivation for what we're going to be discussing today. Also, at any point, if anyone wants to ask a question, feel free to. And if I miss it in the chat, someone can just interrupt and let me know. Um, But so the idea is that we want to take the gene sets from all the CFDE gene sets and interpret them in such a way such that one, they're easy that we can make statistical inferences based on the data, but also we can put it in this format where people can actually using very natural language query the data and get insightful information from it without having to do complicated joins and so on. So I'm going to kind of just take a step back and just begin with some basic motivation. So there's a ton of data, even just in CFDE. And the problem right now is they're sort of siloed. So if you want, for instance, to connect, for instance, I use the example of a chemical response to exercise response in two different parts of the data. Generally what you do is you have these large data sets, you join them if they're in spreadsheet form, and then sometimes this can be gigabytes on gigabytes, and then you have this really, really complicated data structure that you have to go through and calculate and find if there's any connection. And that's often just a, a stumbling block for many people. And so one way to try and improve this problem is to put this into a different format. So um, I'm just going to briefly go over what a knowledge graph is. I'm assuming many people already know, but just for people maybe who are not as familiar, a standard graph is just a set of vertices and edges. And generally speaking, they're relatively semantically poorly enriched. They don't have that much information. So edges connect to vertices. The vertices may represent something, the edges may represent something, but generally speaking, very often there's not much more content. What a knowledge graph is not is taking the same kind of structure. So you still have vertices, you have nodes and edges that connect them, but now you enrich them with more semantic content. So you'll have, for instance, one node represent a subject. They'll be linked via some predicate to some object, and this is extremely general. So a standard example people use, for instance, with knowledge graphs is if you have if you want to model, for instance, the dynamics of a social network, you can have nodes represent things like people or groups, and then you can have predicates represent friends with or doesn't like or and so on. And you get this really rich semantic structure which then, because you have it in this format, so you have predicates and you have the things that hold between the predicates hold between. This allows you to actually make logical inferences. So you can use essentially first order logic, and you can say, well, if this predicate holds between this subject and object, can I then make some further inference about downstream what possibly may hold to the object that connects via another predicate to another object, and so on and so forth. And so unlike the previous format I was mentioning, knowledge graphs can actually make deductions that are not necessarily in the original database. And so this means that you can, without just having to look at a massive spreadsheet, you can actually find information that may have not obviously been there by just scanning it. And another thing that makes them really useful is that they're often standardized with respect to the identifiers and formats. So as long as you follow the specific identifier, you can actually link knowledge graphs that are extremely disparate. So for instance, and I'll talk about this a bit more in the talk, but you can take knowledge graphs for things like biomedical data, and you can combine them and make inferences with a knowledge graph about climate or about a social network. And so that's one of the motivations, and I'll talk about this on the next slide for this project, this Proto-OKN project, which is basically a federally backed initiative where the idea is to get people to upload a bunch of different knowledge graphs that are rich and dense with information. And the idea is that once you do this, you can put them up onto a database, and then you can create this ecosystem where you can um, query these different knowledge graphs and across different knowledge graphs and make connections that may not be obvious. The obvious, one obvious reason you might do this is because knowledge graphs are really amenable to AI, especially contemporary large language model techniques. So instead of, again, having to go through a massive database and having to find merging a bunch of things to try to find specific information, you can query knowledge graphs. And again, I'll talk about this a bit more with an LLM, and it will give you back an answer based on the semantic content I was mentioning in the previous slide. And so the project is meant to harmonize different data sets and put them all together into one place. And then people who are interested can then query these databases, these knowledge graphs rather, and make inferences. Um, so to specifically talk about the knowledge graph that we're working on, we're going to be it's going to be a relatively large one. So it's going to include 7,000 traits, 20,000 genes roughly, and 4,000 mechanisms. And I'll explain more about what these are and where they come from. But the idea is that we want to take all of this massive amount of data that Noel was talking about and um, condense it into such a way that it's much more easily queryable. And so the way that we do this is that each uh, we take all these gene sets, we run specific methods on them, and I'll talk about these more in depth in a little bit, and then we use the outputs of these methods as edges between the different nodes. So in this graphic, the trait, gene, and mechanism are the nodes, and then I'll explain just in a little bit what these specific edges would be. Um, and then from there, we can then ask queries that would basically be impossible to do if we were doing it in the standard way. So For most of this talk, I'll be talking, I won't be talking as much about gene sets, although gene sets will likely be also included in the graph, in the knowledge graph. But right now, I'm going to be mostly focusing on trait, gene, and mechanisms. And so each edge by itself is trying to capture something like what is the level of association between a given pair of nodes. And for each one, because again, we're working on a knowledge graph, it doesn't have to be the same predicate. It can be different predicates. In fact, they are. So what links a trait and a mechanism and what links a trait and a gene will be different. And I'll expound on this in a bit. Um, but in general, that's going to be sort of the next few slides where I try to explicate what exactly is being connected because intuitively there's some maybe kind of naive way to do this. So for instance, a kind of trivial thing you could do is say, well, I have a bunch of gene sets, and then I can construct a predicate where for a given gene, whether it's in the gene set or not. And that's been done, and you can do this. But we generally want to make uh, more informative predicates, things that are not, as obvious. And so the Flanic Lab has developed several methods, statistical methods, to make inferences about what credence we should assign to different trait gene and trait mechanism and gene mechanism pairs. And that's sort of the next slide. But just really quickly, for the sources, the traits come from rare diseases in the Orphanet database and common traits in the NHGRI Association of Function Knowledge Portal and the NHGRI GWAS catalog. And then the gene sets obviously come from all the CFDE gene sets, and they can also come from the MSigDB and MGI databases. Um, and so the first connection that I mentioned is the trait to gene connection. So obviously, presumably, everyone's familiar with what a trait and a gene are, but how they're connected is not so clear. And so there's a method that's been developed, and Noel mentioned this, called Pigeon, which is short for priors inferred from gene annotations. And the basic idea is we want to construct a Bayesian method that calculates relationships between gene sets and traits, and then relates them via something called support. So basically, if you want to think of it in terms of like the Bayesian mindset, you basically start with some assumption, some prior about how much a gene and a trait, for instance, are related. So you might think, my credence that type 2 diabetes is related to this specific gene is this number. And by incorporating a ton of annotation data, you construct this Bayesian method that updates your beliefs, your credences, so that you get a more nuanced notion of what kind of support you should expect between a trait and a gene. So, for instance, you may think that upon looking at a bunch of annotation data, I've learned important connections that I didn't realize before, and so I should increase the credence with which I believe type 2 diabetes is related to this, specific gene in this example. Um, and so what's interesting is it actually gives you several different statistics. So it gives you between a trait and a gene, it'll give you something called an indirect support and so a direct support, and those basically just map to how much support, again, how much support you have with for a trait being connected to a gene based on specific annotation data versus annotation data from things that are not specific but can still be incorporated. And so what Pigeon does is it jointly infers which genes are trait relevant and then which gene sets will predict the trait relevance. And so I won't go too much into the math here, but the basic idea is you construct a statistical model where DI is basically telling you whether or not some gene is related to a specific trait, D, for instance, and you have some latent variables like Y and betas, and you do essentially a Bayesian regression where you start out with some baseline prior like pi, and then you update the belief based on annotations, which are weighted so that you want to control for, like, for instance, if there's extreme oversampling with specific annotation, you want to control for that. So you have certain terms that try to make it so that certain annotations will dominate. And then you use a certain prior because you're doing a Bayesian regression, which is the spike and slab prior. And then you continuously run this, and by the end of it, you basically have this large matrix where for every trait across every gene, you learn some you get some direct and indirect support in terms of log BF and prior, and then also their combined score. Um, so by the end of this, you have a bunch of information from the previous slide that connects the traits to the genes. But now we have to also connect, we have to find some predicate that connects the traits and the mechanisms and the genes to the mechanisms. And for that, what we use is the next method, which is Eagle. And so the idea behind Eagle is it's using something called Bayesian non-negative matrix factorization. And so the basic idea here is that you have after you run Pigeon, you have this a large amount of data, and given the genes and annotations you have from it from a Pigeon, you can using the genetic support probabilities, you can calculate a decomposition. The same way you can do decomposition with a bunch of methods, but what's nice about Bayesian non-negative matrix factorization is it's very, very interpretable, unlike other methods. And all the basic idea, the constraint on it is that you need to have a matrix with all positive values, and that's a given here. And so you then essentially the basic idea behind it is that you take a large matrix of data and then you decompose it into two smaller matrices such that with the constraint I mentioned, such that when you multiply them, you get the original matrix. But what this actually does is because these are oftentimes lower rank approximations, you get these latent factors, these mechanisms, which try to capture these underlying mechanisms that are happening that explain the data that we were previously mentioning. Now, kind of off the like once you run this method, you don't actually get a list of mechanisms with like nice names. That part is usually done after that's the LLM part of the Eagle, where you have these latent mechanisms that are generated from this method, and then from there, like for instance, for a given trait, you may have a number of genes, a number of gene sets, and from this information, then you can query an LLM and say, what is the most likely mechanism that's being explained by this set of genes and gene sets and so on. And so after you run Eagle, you get these latent mechanisms, and they tell you basically the probability that a gene participates in a specific mechanism, as well as the probability that the annotation can actually quote unquote capture a mechanism. And so all of this information is encoded in these edges I mentioned in the knowledge graph. And what this allows you to do is it allows you to ask questions that you otherwise wouldn't be able to ask. So actually, let me, yeah, so before I actually get to this, I wanted to show just a quick demo of what I'm talking about because it's sort of maybe a bit abstract right now. So let me just show some examples. Um, okay. So this is a example I constructed, trying to use more or less the same um, uh, uh, same icons as the slides. But basically, here's a very, very small subsection of what this kind of knowledge graph visually would look like. So, and I'll zoom in on more specifics. But basically, the blue stars represent traits. The green dots are genes, and then these are actually gene sets. And so the strength again, like I mentioned before, the I originally actually tried doing this with just one gene and or sorry, one trait and then all of the genes associated to see like the different strengths of all the associations, but that was enormous because each trait has an enormous amount of gene associations. So what I did here instead was just take four genes, mostly at random, or sorry, excuse me, four traits, mostly at random, and then pick the top 30 genes and gene sets associated. And I was actually very surprised because that's a really strong cutoff. And yet, in a priori, it already captures a lot of the associations you would expect. So, for instance, the traits that were just picked, and this will sort of make sense, is this one is a GCAT trait is systemic scleroderma, which is an autoimmune disease. So it's sometimes referred to as systemic sclerosis. And then this one is lupus. And so just even with this enormous truncation, the data is actually already capturing the fact that these two traits should share more genes in common than the other two, which are Barrett's esophagus, which is when people have chronic reflux, often the esophageal lining becomes damaged, and over time that can increase risks of certain cancers. And then this one is AFib flutter, so atrial flutter. And as it kind of expected, these combine quite well because they're closer together in this kind of latent space. But what's interesting is if you zoom in, each edge here denotes, if I can actually, if I can actually show it. Um, Yeah, so each one will show you different scores. So, for instance, this one is showing you the previously mentioned direct and indirect support scores between a trait and a gene. And these are showing something called betas, which I didn't go into too much detail, but basically that's one of the latent things that are calculated in the pigeon model. And what this tells you is basically, given a gene, how strongly associated is it with a specific trait? And what's interesting is that in this demonstration, despite again this enormous amount of cutting out a bunch of genes, there's a lot of connections between scleroderma and lupus, and this actually makes sense given that there are um, the underlying mechanisms are often very much linked. So, for instance, there's people who have undifferentiated connective tissue disorders where basically it's not clear whether they have lupus or they have a scleroderma, and it's only later until it becomes separated. And so the fact that they share a ton of connected pathways and genes makes sense. But what I was kind of surprised by is that it also captured several gene sets and genes between Barrett's esophagus and scleroderma, which I was kind of surprised by. But upon looking at the literature, there is actually an indirect connection between the two, in the sense that one of them can cause extreme chronic reflux, and over time, because of other issues, it can cause Barrett's esophagus. So this is actually linked, but the fact that it can capture this, even again with these enormous restrictions, is kind of surprising. Um, and so this is again just a very small demonstration of the entire data set, but these kinds of knowledge graphs would be what we're uh, trying to construct. Now, this is not exactly one to one for a few reasons. It's missing some details again because if you include everything, I've tried a few different versions and it would just crash my tab. But for instance, in one example is that in generally knowledge graphs, you don't when for each edge you have one associated predicate. So if you want to have all three, you generally construct like a kind of a hidden node between a gene and a trait, and then you associate these with that. But those are all largely immaterial to the general point. But so the idea is that once you have something like this, you can then query the graph in a way that, again, is very, very difficult otherwise. So you could, for instance, ask pretty straightforward questions like, what are the top 10 genes most strongly associated with lupus, for instance. You could also ask questions that are much more indirect. So you could say, can you find specific mechanisms that may be shared between, say, lupus and Barrett's esophagus? And so the possibilities are endless. And I give some further examples. Let me pull it up. I give some further examples here. So, Sparkle queries are, by the way, the way that you, generally speaking, try to query the graph. And unfortunately, knowledge graphs, as they are kind of by default, you can't just ask via natural language. You can't just put it in plain English and say, oh, what are the top 10 genes associated with this trait, or what explains this mechanism between these two gene sets, and so on. But um, what you can do, and one of the reasons that knowledge graphs are very popular right now, is that current AI models are extremely good at basically taking natural language inquiries and then turning them into Sparkle queries that you can then use on the knowledge graph, and then the knowledge graph can give you back information, again translated in such a way that is just natural English. So, for instance, I give some examples of Sparkle queries that you could give. So, for instance, you could ask, what are the most strongly associated genes for type 2 diabetes? And then again, you can ask more specific questions like, I want anything that has the pigeon combined score that's greater than 10, or the number, it can be any number. and then it can translate it into a Sparkle query. And then the same thing, you can ask, are there any latent biological mechanisms that Alzheimer's disease and type 2 diabetes share? And again, same thing. And this is actually really just like the tip of the iceberg. You can ask very, very complicated questions, and you can get uh, really, really interesting results doing it this way. And so I do want to just briefly talk about a little bit of the architecture. So one of the benefits of the Proto-OKN project is that they have this architecture set up where basically, like I said, you can give a natural language query, and there's an MPC client, MCP client, and then it basically takes the natural language, it uses LLM there's a set of MCP tools, and then from there, it's able to get some understand the context, query the graph, convert it back into natural language, and then just give you a straightforward answer. Um, so I do want to also, if time permits, I do want to also try showing another demo.
Yes, please feel free to do that, Luca.
Okay, cool. Let me just pull it up. Mm, okay. So another thing that we can also include in the data, and let me see if this works,
Perfect.
is
Yep.
yeah. So I mentioned kind of I kind of again I'm kind of glossing. So if anyone has any questions, feel free to ask because I don't want to get into too much of the into the weeds of the Eagle method. But one thing that's uh, possible with Eagle is that I mentioned you can run this BNMF, this matrix factorization on the original large matrix, and you can get these latent mechanisms. But you can actually keep repeatedly doing this. So you can do it for one round, and then you can get these factors, and you can run it again and get even like coarser snapshot of the data, and you can keep going. And so again, some of this is somewhat early on, but we found that by doing this with for several iterations, you can actually get a hierarchy of different levels of organization. So you start out very low with mechanisms that are very, very specific. So you get mechanisms. So for instance, this is just some example of some data where you have for a given factor, you have really strong the top gene drivers, and then you have these annotations and you have these mechanisms. But you can actually keep repeatedly doing this, and as you do this, you start moving from really, really specific mechanisms like very specific cytokine, regulation. You move up to things like general physiological systems and organs, and you can keep going up higher and higher. And so, what this allows, and this is just an example of this in action. What this allows you to do is basically ask not just very specific questions like on this very specific hierarchy level, how is this trait associated with this gene? But you can actually ask if you incorporate this data things like, oh, how does this trait and this gene relate to this specific higher order meta factor or meta mechanism? And it can make inferences across different hierarchical levels, which is really, really useful. Um, and so you can actually, and this is again somewhat early, but you can find that there's certain mechanisms can actually cluster themselves into a latent space, and then you can make inferences about meta mechanisms that then you can infer data from lower level mechanisms, and so on. Um, so let me just share again. Right. So this is just a very, very short, simple examples you can construct using the knowledge graph. But this actually doesn't go into at all really any queries you can make with other knowledge graphs, right? So one of the things I mentioned is that you can, in addition to making queries on your own knowledge graph, you can use really disparate ones and try to make inferences between those two. So I give two kind of very simple examples. And again, I think because there's so much cross queryability, essentially, since everything is on the same standard, you can really, really take this very, very far. But as way of an example, I was thinking that if you wanted to, for instance, understand how specific physical protein-protein interactions occurred, given there's a Pro-KN is an example of a data set in another data set in Proto-OKN, you could then query both graphs so that you start with a trait and then you ask how do the specific PPI data explain the biological mechanisms from a specific gene? And so you can link protein-protein interaction networks with the data we have and try to make inferences that way. That's sort of a very natural way of doing it. But you can also even go really far, and there's data sets from many different disparate fields. So you have data sets about climate, about certain farm data, and you can ask questions like which genes linked to respiratory mechanisms in our graph are highly expressed in places with poor air quality, and all of those are perfectly kosher to ask, and you can make really deep inferences that you otherwise wouldn't. Again, because you'd have to join massive tables of data and then trawl through them and try to figure out if there's any connection. Um, so one thing I do want to just briefly, I kind of skipped over this a little bit, but um, one thing that we could also do with Eagle is um, because Eagle is it's not a priori setting a strict amount of mechanisms. So for instance, when you run Eagle, you use a specific technique where so essentially prune mechanisms so that there's no a priori required amount when you start. So you're not biasing the model very strongly by saying, okay, you have to condense all of this into 10 mechanisms or 20 mechanisms. You start with a high number and then it prunes based on some Bayesian reasoning until you're a smaller number. And so the idea, though, is that you can actually attempt to take these mechanisms and then try projecting certain data onto the mechanisms so that you can actually make further inferences like if I have this specific disease state, for instance, um, and I have 20 mechanisms, and these are all labeled via LLM, what percent of each mechanism contributes to that disease state? And if you have good labels for the mechanisms, you can literally say, you can make inferences like, um, this specific disease is mostly driven by this specific mechanism, but not driven by this mechanism. And each mechanism then is further queryable into a set of gene sets, a set of top genes driving it, and so on. And so you get this really rich structure that you can project onto when you use these um, mechanisms. Um, and then I think there's, yeah, so um, in terms of the status right now, so we have the all of the CFDE runs finished. We have the model in Turtle and then RDF format as well. And so we just need to, do a double check of the specific data, but we will be updating and uploading the graph soon. Hopefully, the end of this week or maybe early next week. And then in the future, the goal is to have it, again, fully integrated with this LLM MCP server so that people can test it and just ask really simple and plain English questions that then the model can give you back an answer for. And we'll release all the data and code and documentation, of course. And if you want more information, I've also included some links. But that's it for me. If there's anything else, feel free to ask.
Thank you, Luca. Um, So I will take a look and see if there are any comments in the webinar chat. In the meantime, if you have any questions, please feel free to raise your hand. We can take you off of mute, or you can use question and answer. Either way works.
Luca, that was excellent. It was really great.
Thank you.
It's actually really exciting, I think, time for us. So the what Luca also indicated is in addition to our CFD reveal knowledge graphs, there's like four other knowledge graphs in the CFD ecosystem itself that we'll be hanging ours on and connecting to so that we can query all of the CFD data as well. And then also he mentioned with the Proto-OKN, it opens up an entirely new universe, which is very cool. And it kind of like to someone who's like a biologist like me, you're like, what? I can now do these things I never could before. So it's also like when you see that I saw your demo, I'm like, there's this hairball. But then when you start to tease out the science, it really does start to connect to things that, folks who are in the work and then the wet lab actually find relevant. So that was excellent. Excellent. What questions do you all have or thoughts? Or like you're like, how am I going to use this as a user? Or what are you guys going to do to make my life easier when you release this thing? Any questions along those lines? All right. The question in the chat was excellent about pilot funding. I think Julie answered it, but just for those of you who know, this is actually a really great opportunity for folks outside the CFD to get funding to work with us. And if you are an existing CFD funded investigator, I think the guidelines are if you're a PI, you cannot be on the RO3s. But you could be like a member of your lab could be on them, so someone else was prime. So there are very cool opportunities for if you're an existing partnership or a CFD awardee, you can sort of allow other people in your trainees or folks in your lab to be part of those if you're so interested. And if you have any questions about that, you should definitely reach out to the program officer or to us, and we can put you in contact with the program officer because they can answer the best questions. But that's what we know from just being part of CFD. Other questions you might have? Okay. We will make this available, as Mackenzie said, as always, if you want. And then tomorrow, if you want to hear more stuff or tell your friends, we're going to be talking a little bit more broadly about Reveal. So Reveal is the current user interface that you can make natural language queries on top of the knowledge graph that Luca mentioned today. So it gives you, it's our sort of first blush at that. And what's cool is we got some benchmarking to share. So we've actually said like, hey, if I had Reveal 2 years ago when I was writing this paper and I had the specific query I wanted to make and I typed it into Reveal, would it have either confirmed some of my findings from my paper or actually add additional information? And we've done some early benchmarking on 10 papers from the literature where we've actually been able to determine that Reveal does a pretty good job in many cases. So I'll reveal that tomorrow to you if you want to join at our webinar time for CFD. And you also hear more about CFD more broadly and our other activities to connect knowledge graphs more readily across the ecosystem. So that'll be tomorrow's soup du jour and a new release we have under the Knowledge Center, which will help you navigate that site we showed earlier. All right, Mackenzie, I'll hand it off to you. Thank you,
Yeah.
Luca.
Thank you, Luca. Thank you, Noel. We greatly appreciate it. As a reminder, these are going to be more frequent. We understand that this is a new cadence, so thank you for those of you who are able to join today. Our next meeting will be in 2 weeks, and we are very much looking forward to seeing you all there. Again, if you have any questions, concerns, please reach out to me. This webinar will be available on YouTube, and I'll send this out. So please feel free to forward to any of your colleagues who are unable to join today. So thank you again, everyone, and we greatly appreciate you taking the time to join us today. So thank you. Bye.